I want to welcome everyone as you're coming in. We'll be starting in about 2 minutes. We're just going to let everyone come into the room first. Be just a few more minutes. Just welcoming everyone as they arrive. We'll be starting in about 60 seconds here. I'm just going to let everyone get into the room first. All right, I'd like to welcome everyone and thank you for being here. We are going to get started right on time today. I just want to let you know who I am. I'm Susan Boas, the Executive Director of the PANDAS Physician Network, and we really appreciate you joining us for our webinar today, The Role of Adaptive Immunity and Genetic Risk Factors in Vascular and Neuronal Dysfunction in Post-Infectious Autoimmune Encephalitis. This is presented to us today by Dr. Dritan Agalliu. And before we begin, I'd like to just give a reminder to the attendees. There is a chat function where you can ask questions to our presenter. Please remember that those questions today should be research oriented, as this is a research presentation and relevant to the presentation topics. Additionally, this webinar will be recorded and it will be added to the PPN website if you would like to go back and review it after this session. If you will be requesting CME credits, that information will be sent to you after our live stream. We'd like to express our appreciation to at Point of Care, who has made the accreditation of today's webinar possible. And now we'd like to welcome our presenter, Dr. Dritan Agalliu. He is an associate professor of pathology and cell biology and neurology at Columbia University's Irving Medical Center. He is a neuroscientist with 20 years of research experience in developmental, cellular, and molecular neuroscience, and has specialized in the interaction of brain and blood vessels in healthy and as well as diseased central nervous systems. Dr. Agalliu also has a passion for the visual and performing arts and classical, jazz music, and dance. Dr. Agalliu, I will turn this over to you.
Thank you, Susan, for that wonderful introduction, and also for inviting me to share with you today our research. So I'm going to share the screen right now. So what I would like to tell you today is two major stories, one in our animal model and the second one using human data to really try to understand the role and the mechanisms of the adaptive immunity in regulating, in controlling the vascular and neuronal dysfunction in PANDAS PANS, but more broadly speaking, I want to think about this in the context of a post-infectious autoimmune encephalitis. And here are my disclosures. The mouse work that I'm going to present in the first half of the talk is funded by the National Institute of Mental Health, R01, and also, whereas the basically the human work on the cytokine profiling in PANDAS PANS children that I'm going to present in the second half was initially funded by a grant from the International OCD Foundation. We're very, thankful to them, and a Camper Basic grant from Columbia, as well as funding from the PANDAS network. And the genetic studies are primarily supported by funding from PANDAS network. Obviously, a lot of this would not have happened at the beginning without a tremendous amount of support from Newport Equities. So I want to broadly start this webinar by discussing a little bit about autoimmune encephalitis. So what is autoimmune encephalitis? autoimmune encephalitis is, broadly speaking, are a large number of diseases where basically antibodies that normally circulate into the bloodstream and are excluded from the central nervous system, enter the central nervous system and begin to attack neurons. And basically they recognize on the surface of neurons either receptors for neurotransmitters or synaptic proteins, and they trigger the internalization or removal of these receptors from the surface of the neuron, leading to abnormalities in the function. And these are manifested generally with either neurological or psychiatric sequelae. Probably a lot of you are familiar with the book Brain on Fire. And I think that that's a beautiful example of how autoimmune encephalitis can affect the function of the people. So obviously, these diseases came to emergence or they became very clear where basically Dalmau and colleagues in 2009 first identified antibodies that recognize basically a receptor for synaptic, for a neurotransmitter, an excitatory neurotransmitter termed glutamate. And this is the NMDA receptor. But over the course of 10 or 12 years since the initial discovery and description of this, we now know that indeed antibodies that recognize other synaptic proteins, so other receptors for the glutamate, AMPA receptors, and GluR5, but also for the inhibitory interneurons have been identified. And obviously, my lab focus is really on the, our general interest is on the blood vessels of the central nervous system. And we're very intrigued to understand how do these antibodies that attack neurons in these diseases enter the brain, by which mechanisms and how they induce disease. Now, when we think about autoimmune encephalitis, what has been predominantly described has been really that the primary cause of the generation of these antibodies is basically following a ectopic tumor in a tumor setting, or what we call the paraneoplastic syndromes. Basically, in the context of this, a tumor found in the body that has some properties or characteristics of the nervous system triggers an immune response by the body. And during this process, a lot of the antibodies that are generated targeting the tumor tissue, including a nervous system component, then circulate in the body and then begin to attack the central nervous system. However, the autoimmune encephalitis, either broadly or more restrictively, also can occur in the context of patients who don't have tumors. But also, it more recently has become clear that also it can occur in the context of post-infectious disease. So, broadly speaking, what I'm going to focus today is basically post-streptococcus sequelae that attack the basal ganglia that collectively fall into the pseudonym scoria or PANDAS. But also probably what most of you are familiar, we're living through a pandemic, and there has been a lot of description that indeed following COVID infection, there have been neurological and psychiatric sequelae in a lot of patients. And so this really brings to the attention the concept that in fact, many of these diseases targeting the central nervous system could be due to a post-infectious effect. So regardless of the target, whether the target is basically the glutamate receptor in autoimmune encephalitis or the dopamine receptors in basal ganglia encephalitis, obviously the key question that we've been interested in my lab for a long time has been on how do these antibodies gain access to the brain? And this is an important question to consider because the blood vessels in the brain are somewhat unique compared to blood vessels in other organs. And this uniqueness is termed the blood-brain barrier, a property of the vessel described by Ehrlich and colleagues at the beginning of the 20th century. And this is illustrated in this schematic here. If you take a dye that binds to albumin, this is a blue dye, and you inject that into the bloodstream, you can see that as it binds to albumin, the dye basically will leak out of the vessels from other organs such as the kidney and the liver, and will turn these organs blue. But in the brain, the dye is unable to leak out of the vessels, and so the brain remains clear, white. And so we do know that this property is not because the dye cannot stain the tissue per se, the brain tissue, because you can see that in the context of diseases where the blood-brain barrier is damaged. But this is predominantly because basically the blood vessels in the brain contain this property termed the blood-brain barrier that is essential to maintain the homeostasis of the central nervous system and to limit the entry of pathogens, immune cells, and drugs. So when we think about autoimmune encephalitis, broadly speaking, and how do these pathological antibodies can enter the brain, there are potentially two ways in which one can happen. One of them described here is when the cell-cell junctions between cells are destroyed. And so the antibodies can cross basically this barrier and enter the brain. And the second mechanism is when basically the antibodies recognize cognate receptors on the surface of the blood vessels, and then use these to basically cross the blood vessels and enter the brain. Now, basically, we're interested in trying to understand this in the context of post-infectious sequelae of the central nervous system. And probably the best understood so far and the better characterized is the post-infectious sequelae by group A strep infections. So group A strep, or Streptococcus pyogenes, is a gram-positive bacteria, is the common pathogen for the sore throat that we all experience. It basically is a very common pathogen in children because children experience a lot of sore throats, but has been implicated in a large number of autoimmune sequelae, including rheumatic fever in the heart, glomerulonephritis in the kidney, scarlet fever. And then in the CNS, the more accepted and obviously well characterized is the neurological sequelae of the rheumatic fever, described by Sydenham, in the 19th century, and it's termed Sydenham's chorea. And then in the 90s, Susan Sweden and colleagues at the NIMH, as they were studying Sydenham's chorea, they basically identified a subset of patients that were manifested more predominantly with acute psychiatric sequelae rather than neurological sequelae. And basically, she coined the term PANDAS for pediatric autoimmune neuropsychiatric disorders associated with strep. But in the context of these diseases, what is very clear from a lot of work, including Madeline Cunningham's work, is very clear that indeed an immune response that targets the pathogen basically becomes aberrant. And so basically either antibodies or T cells that are generated to fight the pathogen then erroneously begin to recognize basically proteins that are normally present in the tissue, in the heart valves, or in the brain, the basal ganglia, leading to basically disease. When we think about Sydenham's chorea and PANDAS, there are obviously a lot of similarities, even in the initial description that Sydenham had. He describes obviously a lot of psychiatric symptoms, including OCD-like symptoms, emotion liability, and contamination fears. But obviously there are very clear distinctions between the two diseases. So what we've been interested in trying to understand a little bit the mechanisms of disease. Obviously this is kind of difficult to do in humans, but basically we are experimental biologists in my lab. And we like to develop animal models for the disease so we can understand the disease better. And so we have been working over the past 10 years since I started my lab to model and develop a disease model for post-infectious autoimmune encephalitis in mice. And so in this experimental model that we initially developed in collaboration with Patrick Cleary at University of Minnesota was basically to do intranasal infections with group A strep weekly in young mice to mirror what actually happens in humans. And then after several repeated infections, we can analyze what happens to these animals. And so when you do repeated infections with group A strep, in mice also become sick, a little bit less than humans, but they do become sick because group A strep is not a mouse pathogen, it's a human pathogen, but they still, you can see that basically the group A strep is located in the nasal cavity, just like in children. And then it's surrounded really by a basically large number of immune cells. Visualizing here are the T cells or the T lymphocytes. And the cells are generated to basically predominantly fight the infection. But basically the cells are predominantly localized in a particular region of the mouse nose called the nose-associated lymphoid tissue, which is the equivalent to the human tonsils. And so given the proximity of this tissue to the basically olfactory epithelium, which is basically where a lot of the nerve cells that are important for the sense of smell are located, and their cell bodies are located there, and they send their projections into the brain. We actually initially asked whether these T cells that are present in the nose can use this as a route to travel to the brain. And what we did publish in 2016 in collaboration with Patrick Cleary was that indeed this was the case. So if you compare basically the naive mice with mice that have 2 infections versus 5 infections with group A strep, you can see that repeated infections with group A strep lead to basically an accumulation of immune cells, lymphocytes in the brain. But their distribution is somewhat unique. They're very high in the more anterior parts of the brain, and in particular this region, which is the olfactory bulb, where neurons that are olfactory neurons that get the information about the sense of smell send their axons into the brain, their projections into the brain. And their number is reduced somewhat in other more posterior regions, but yet it's still significantly higher than either in naive mice or mice that had 1 or 2 infections, suggesting that multiple infections with group A strep are really important for driving basically an immune response or infiltration of immune cells into the brain. And because this paper is published, I'm just going to illustrate in a cartoon what actually we have seen. Is that we have seen that following this infiltration of the immune cells into the brain, they break down the blood-brain barrier by basically disrupting the cell-cell junctions and promoting transport of antibodies. These antibodies now enter from the bloodstream into the brain. Basically, in addition to this, the innate immune cells of the brain called microglia become reactive, they become activated, and this leads to the loss of neuronal activity. So because the T cells, because basically in this initial study in 2016, we saw a nice correlation between the presence of T cells with basically the pathology, we questioned whether indeed Th17 lymphocytes that generate this cytokine called IL-17 are required to induce the pathology in our mouse model. And so to do that, we used a genetic mutation in mice that basically eliminates the function of a key protein. This is a transcription factor that is essential for generation of Th17 lymphocytes. And so in this mice, basically these cells are generated either at low or no numbers. And so a former graduate student in the lab, Marianne Platt, what she found is that basically after repeated group A strep infections, what you do see is that indeed, whereas in the wild type mice, there is a large infiltration of immune cells, in mice deficient for Th17 lymphocytes, the number of these T cells is reduced. And when we do look at the different populations of lymphocytes that enter the brain, we can separate them by expression of different secretory cytokines into Th17 and Th1 lymphocytes. We can find that indeed the number of Th17 lymphocytes is reduced, whereas the number of Th1 lymphocytes is increased. So we're in a perfect situation to really examine what are the consequences for the brain pathology when you don't have Th17 lymphocytes. And what I want to emphasize here, the reason we focused on Th17 lymphocytes is that these lymphocytes have been implicated in virtually every single autoimmune disease that we know today, and they play a critical role in breaking down the blood-brain barrier and inducing loss of myelin in multiple sclerosis and its animal model experimental autoimmune encephalitis. So there was a logical, reasoning for us to focus on these cells, given their pathological implication in other diseases, including CNS autoimmune disease like multiple sclerosis. What we did find in these mice, though, is that following multiple infections with group A strep, whereas in the wild type mice, you see a massive infiltration of antibodies from the bloodstream into the brain. You can see that these are absent in mice that have never seen infection with group A strep. Their number is reduced significantly in the mutant. And the amount of the antibody. And actually, one of the reviewers for our paper did ask us to repeat this experiment by injecting intravenously IgG that was labeled with a red fluorophore. And within 30 minutes of the injection, we did see that basically that antibody was able to enter the brain pretty rapidly in wild type mice after multiple group A strep infections, but not in mice that had not, where the Th17 lymphocytes were not able to be generated, suggesting that indeed the Th17 lymphocytes are really critical for damaging the blood-brain barrier in the animal model. The second feature that we looked very carefully was the state of microglial activation or macrophage infiltration, because basically this is an indication of the degree of neuroinflammation. And for those of you who are interested in human studies, the degree of microglial activation is also a way by which you can measure neuroinflammation even in patients using PET imaging. And so basically in this case, what we did see is we measured the degree of microglial activation here by looking for the expression of a lysosomal marker, CD68. Lysosomes are organelles that are required for the phagocytic activity of the cells. And so you can see that basically the amount of microglia or macrophages that have CD68 expression is really high following group A strep infections, and it's reduced in the Th17 deficient mice. So finally, what we did is a functional study where basically now we wanted to test the ability of the mice to smell. So these were mice that are alive, where we basically inserted electrodes to measure the activity of the neurons in the brain. And then the mice were presented with different odor smells, so we could measure whether the mice were able to smell or not. So obviously, what I'm showing here are mice that have never received infection, and on the left are mice that have Th17 lymphocytes, and on the right are mice that lack Th17 lymphocytes. And you can see as you present different odors and increasing concentration, you see this little spike or peak. This is basically the activity of the neurons, suggesting that the neurons are active and they begin to fire what we call action potentials in the presence of an odor. So suggesting normal neuronal activity. But things look different in the context of group A strep infections. So you can see here in the left, in mice that basically have Th17 lymphocytes, no matter how much we increase the concentration of the odor, we see no response. Just basically, this is simple noise. However, in the mice that are deficient for Th17 lymphocyte, there is a rescue in neuronal activity. And so this beautiful data published last year by my graduate student, Miriam Platt, suggested somewhat complicated model, but I want you guys to focus on the bottom panel here. So whereas in the top one, I showed you that in the presence of Th17 lymphocytes, you have damage of the blood-brain barrier, entry of the serum IgGs from the blood into the brain, activation of this innate immune cells called microglia, and loss of neuronal activity. In the absence of these cells, you rescue all these phenotypes. So you basically have less antibody entry into the brain, the microglia look less activated, and then obviously there is a rescue in neuronal activity. And so obviously one of the questions that we have been interested recently in trying to understand is whether indeed the molecules made by the Th17 lymphocytes, IL-17 or GM-CSF, do these things alone, or do they involve basically an intermediary population called the microglia? And also trying to understand what type of inflammatory molecules the microglia or the macrophages that infiltrate the brain may secrete to basically drive the vascular pathology. And so in order to do that, a current graduate student in the lab, Charlotte Wayne, has performed single-cell RNA sequencing. So basically in this approach, you take the brains, you dissociate them into single cells, and then using sophisticated computational approaches, you assign cells identities and you look for basically changes in their gene expression as a readout of basically what is happening in the brain. So you can see here, these are what we call Tisney plots, where you can see in the left here, the identity of different cell types that Charlotte is able to identify. So she can identify the neurons, the microglia, the endothelial cells, the astrocytes, macrophages, and T cells. And then in the olfactory, there's also a specialized population called the olfactory sheathing glia. Now on the right here is that same plot, but now it's plotted differently, where in the blue are basically cells that are isolated from the healthy brains, and in red are cells that are isolated from the diseased brains. And one thing to note is basically you can see that the T cells are virtually almost all of them coming from disease state, suggesting that basically you have very few cells in the healthy brain, but these cells are really present in the disease state, which is what we had seen before. So are the macrophages, absent in the healthy brain, or very few, very large number in the disease. But the two populations that show very dramatic shifts in the expression of their genes are basically, one is microglia, where you can see a dramatic difference between the left and the right, the left being basically disease and the right being healthy state. And the second population are the endothelial cells, where you can see a very clear separation in basically their transcriptional identity between the healthy state and the disease state. So based on these gene differences, we can also look for basically what are the genes that are changing. And so when Charlotte did this quantitative and this analysis using a software called Ingenuity Pathway Analysis, she was able to identify that indeed the gene signatures that were highly upregulated in endothelial cells in disease state were basically those that are found in the context of inflammation, no surprise, basically chemokine, cytokine signaling, antigen processing and presentation inflammation. And the ones that most predominantly were downregulated or gone down were genes that regulate the function of the blood-brain barrier. So this is a nice confirmation at the RNA level, at the transcriptional level, of basically those functional studies we had seen before. But what was very intriguing is to try to see, in the context of microglia, what are these cells making in the context of disease? And so what was very intriguing and very, very exciting was that we found that basically the majority of the chemokines that are, or cytokines that are involved in inflammation were made here by either the microglia or the macrophages. So these are violin plots, basically where in the red are cells from disease state, in the blue from the healthy state, and each dot represents basically a single cell. And you can basically, or expression level, and you can see here for several of these inflammatory cytokines, they're basically predominantly made by infiltrating macrophages or microglia with a few exceptions. For example, CCL5 is made by T cells. And if you look at a few more of these inflammatory molecules, IL-1 beta, TNF alpha, IL-1 RN, they're also predominantly made by microglia of macrophages. And obviously this was very exciting and interesting to us because in parallel studies, we had also starting to understand, what is different in the bloodstream between patients, PANDAS and PANS patients versus controls. So with initial funding from the IOCDF and also continuous funding and support from PANDAS network, we had really been interested in trying to understand what are the cytokines and the chemokines that are different between controls and PANDAS and PAN patients with a hope of identifying potential biomarkers for disease, which currently are basically very limited or missing. And so in this study, we were greatly helped with a collaboration with Susan Swedo while she was at the NIMH, where we obtained basically about 14 cases from NIMH and 11 controls. And then 20 cases have been obtained in collaboration with Wendy Vargas, Jennifer Bain, and colleagues in the Division of Pediatric Neurology here at Columbia. So this is basically the average age of the cases and the controls, their distribution by sex, and also basically the source where these sera were collected from. And so this work was actually work of Tyler Cutforth, who is a associate research scientist who has independently been interested in this. And so when Tyler basically used Luminex immunoassay to identify potentially what cytokines were elevated in PANDAS and PANS cases, these are probably the 10 most highly upregulated cytokines that are present in PANDAS and PANS at very high level and very, very low levels in the healthy control. And these are really quite highly significant. And so, potentially this could represent a panel of really significant or important biomarkers for the disease. The other thing I want to highlight to you today is that basically I personally was very surprised to see that a lot of these cytokines that are highly upregulated in patients were the very cytokines that we identified in macrophages or microglia from our animal model, suggesting that indeed our animal model represents a really good model to study this disease because the majority of these cytokines were also highly upregulated in expression in either infiltrating macrophages from the periphery into the brain or in the activated microglia. And potentially they suggest that some of these may also be secreted in the bloodstream. So why is this important in addition of just being a biomarker for the disease? This is important because one of the questions that we had is in the acute phase of the disease where these items were collected, can the sera from PANDAS or PANS patients really affect the function of the blood-brain barrier? And obviously this is difficult to do in culture, but basically one of the approach that we had is we could take endothelial-like cells developed from iPSCs, and we can culture them in vitro in the presence of different compounds to see how they affect the properties of the blood-brain barrier. And one way to do that is that we can look for the ability of the cells to transport things. Basically, in this case, we're going to look for the ability to transport albumin, but also this could be also an indirect assay to measure the ability of the transport for basically antibodies. And the surprise thing was that basically when we cultured these cells, so in the left here is basically cells cultured alone. Here are in the presence of two very well-known inflammatory cytokines, IL-1 beta and TNF-alpha. And then here on the panels A2 and A3 are basically cells cultured with sera from patients, either a male or a female PANDAS and PANS. And the thing to note is right away that surprised us is that the amount of the albumin that was actually taken up by endothelial cells was really high when the cells were pre-incubated with the sera from PANDAS and PANS patients. This is work that Tyler and Nicole Amputi did. And what you can see here is that indeed, in the context, the sera from PANDAS and PANS patients highly upregulated the transport of albumin. Now, given what I told you about the type of cytokines that are present in the sera, we then tested individual cytokines for their ability to do this. And obviously, to our surprise, the key cytokines that are expressed by macrophages and microglia and that were highly upregulated in the sera of patients with PANDAS and PANS were also very significantly able to increase the transport across these endothelial cells, suggesting that indeed in the acute phase of the disease, these cytokines secreted by infiltrating or circulating macrophages could potentially also damage the blood cells. So you could have basically a double-edged sword coming both from the periphery and the brain in damaging the blood-brain barrier in the acute phase of the disease. And so basically, this is just a summary of what I was telling you so far, that indeed several of these inflammatory cytokines that are present in the bloodstream in the context of PANDAS and PAN patients, but also in the mouse model, they're also present on the other side in the brain, seems to really trigger the transport of antibodies or immunes or albumin across the endothelial cells. So in the last 15 minutes or so of the seminar, I'm going to tell you a little bit about our efforts to try to understand another aspect of disease that obviously is very puzzling, but also quite concerning about parents. Obviously, we do know that indeed group A strep infections are very common in children. Excuse me, but one of the big questions in the field for a lot of physicians, but also parents, has been why not all the children develop the disease, even though group A strep infections are very common and prevalent. And so we've been really intrigued about understanding what are potentially genetic risk factors that may drive this disease in patients. And so this work is supported by a PANDAS network right now. And basically the idea here is that you can collect the bloodstream from patients, and then you can do, in principle, two different approaches. So for diseases like multiple sclerosis, Alzheimer's, where you have a large number of patients, then you can do the SNP genotyping and genome-wide association studies to identify risk genes. We don't have this luxury in the world of PANDAS and PANS because we have a limited number of patients. And so because of this, for basically a limited number of patients, we've taken a different approach called the whole exome sequencing, where you sequence only the coding region of the genome, of the human genome, to identify rare mutations, but they can be deleterious for the function of the protein. And so in this case, we are identifying rare cases where basically these mutations may cause loss of function and therefore lead to disease. And then we use computational approaches to really weight the effect that the mutation has on particular disease. And so this has been a great collaboration with Children's Hospital of Philadelphia, Hakona Connerson and Michael Gonzalez. And also the cases have been really collected both from CHOP and as well as COIMC in great collaboration with a pediatric neurologist here, Wendy Park and Jennifer Bay. So this is the description of the cohort. So as I said, we have about 43 cases from CHOP and 27 cases from Columbia. The cases from CHOP were collected retrospectively from the electronic medical records, where basically these were identified cases of PANDAS or PANS, even though there was no code there. And then the prospective cases were collected here at Columbia. And this is the distribution of by age, by sex, and by race. And basically the subtype, the majority are PANDAS and PANS, but we did have 17 patients that had syncytial. And so we were able to basically determine here commonality and difference is basically in these genetic risk factors. And this again has been a great collaboration between Mike and Gonzalez in the laboratory of Hakona Connerson and Tyler Atforth, an associate research scientist here at Columbia. And so the computational approach that we have used here is a little bit complicated, so I'm not going to go into detail, but just to give you a flair about it, is a software called ANOVAR, developed by Hakona Connerson, that basically allows you to to look at the relationship. When you identify this particular mutation, to look at the relationship of this mutation with the gene, whether the mutation is located, in what part of the gene, functional of this mutation, and then to look at basically the conserved region, and then to filter these with other software, basically that are found in other databases. And then once you identify these genetic variants, basically you use a lot of software, approaches to really filter out and identify things that occur, at rare frequency throughout the whole population, and then identify rare, non-synonymous deleterious variants. And I want to point out here, because I didn't mention the controls. Were 5 times more. So we have about basically about 350 controls that have all come from Children's Hospital of Philadelphia. Basically, they have a database there where every child that comes to the hospital, they collect the blood, and they have that, so they have like a lot of patients there that, don't have, so we selected for patients for controls who don't have like autoimmune diseases. Sorry, I haven't updated the number there. And so we, after all of this, you use a lot of filter-based approaches and test to basically identify and weight variance in cases and controls, and then to basically to identify basically the damaged gene variants. So based on this, what I'm sharing with you is basically a list of the top 10 ranked genes from whole exome sequencing using this approach called the gene burden analysis. Doesn't matter very much for basically to identify risk gene in post-infectious BG. And as you can see, there are some genes that make sense. So as you can see, we have an HLA class 2, HLA-DRB5, that basically is an immune-regulated gene. The top candidate gene is GGT5, which is an important enzyme involved in glutathione metabolism, oxidative stress, leukotriene synthesis, and B-cell mediated inflammation. And the second gene is also a gene that is involved in basically process of phagocytosis in macrophages, this infiltrating immune cells. And so what I'm trying to tell you is basically a large number of genes here, maybe 5 out of 10, are genes that are related to the immune system. So keep that in mind, because this is an important finding, obviously. And when you look at the expression of a lot of these genes, a majority of them are expressed in the cells of the immune system, the T cells, the macrophages, the antigen-presenting cells, the microglia as well. Now, we also used a different approach where in this case, we looked for the top single nucleotide variants that showed the most significant difference between in the frequency between the cases and the controls. And to our surprise, again, GGT5 came in the top list. So you can see here, it tells you basically the name of the gene, the chromosome location, the number of the variants, the position, and then it tells you what the mutation is and the frequency. You can see that in cases, even though it is a rare mutation, only 4% of cases had this, it is completely absent in 300 controls, suggesting you that basically, this is highly significant. And again, a lot of these genes where you can see here are emerging that, again, OXR1 and WIPF3 that were in our list before are also emerging here. So we obviously have become very interested in GGT5 because it's at the top of our gene list, and for a good reason, because GGT5 encodes for this enzyme called the gamma glutamyl transferase 5, is a member of a gene family encoding an enzyme that converts leukotrienes, C4 to D4. And of course, for those of you who have interest in immunology and inflammation, we know that leukotrienes are basically key molecules that are made during inflammation. And a lot of the basically non-steroid anti-inflammatory drugs target particularly this pathway. In addition, recent studies in humans have also identified that GGT5 is also important for confinement and homing of B cells to the germinal centers. And B cells are important for generating antibodies. It is known that GGT5 is highly expressed by the tonsils in humans and other lymphoid organs. So one of the questions that we had is GGT5 relevant in our animal model? And so here's what I'm showing you here is our animal model, obviously, like, just to remind you here on the top what's happening in the brain. But when we look at the expression of GGT5 by RNA levels, you can see that in the context of the olfactory epithelium here and the olfactory bulb, which is really here, the levels of GGT5 are highly upregulated, suggesting that indeed there is a gene that we identify from the human genetic studies that is really highly upregulated also in our animal model, suggesting again that indeed the animal model may really represent an important advance to really understand the disease. And obviously we're very intrigued and interested in pursuing our genetic studies to really address the role of this gene in the disease. So with that, I'm just going to summarize that indeed we have been using this genetic approach to really address the role of the genetic risk factors from post-infectious basal ganglia encephalitis using, I think, this computational approach. And we're really interested in taking this last step to evaluate genetic risk factors. But obviously, we've been successful, even though the number of patients has been small, to identify genes related to immune function. And so in an unbiased manner, what we really have uncovered in that indeed PANDAS and PANS is an immune disease. Basically, a lot of the genes associated that cause disease are genes that function in the immune system. And indeed, the majority of these genes function in either T cells, macrophages, microglia, or B cells. And I don't think that we can question based on these preliminary genetic studies. Obviously, we need to increase the numbers. And of course, we're very intrigued about functional studies to really address the role of these genes in our disease. With this, I want to thank a few people. This is the end of the seminar, and I want to thank a few people here. First, I want to thank Marianne Platt, shown here in two pictures. This is my lab, the evolution of the lab. Marianne Platt was a fantastic graduate student who really carried out a lot of the studies published in our PNAS paper to address the role of Th17 lymphocytes in the animal model. A lot of the human genetics, and that work is now being carried out by Charlotte Wayne, a current graduate student featured here. With the help, obviously, of an undergraduate, Sarah Ho. And basically, the human studies are pioneered by Tyler Cutforth. He really pioneered all the genetic studies by setting up the collaboration with Children's Hospital of Philadelphia, and also pioneered all the cytokine analysis. Initially, these were carried out with Nicole Ampati, who was our clinical coordinator, and now they're continued by Michael Glendinin. Obviously, this would not have been possible without collaborators. Initially, the mouse model was set up with Pat Cleary at University of Minnesota, Susan Sweeto for her wonderful collaboration to provide sera for our cytokine study. Obviously, Wendy Vargas, Jennifer Bain, and Shannon Lanny, a child psychiatrist here at two child neurologists and a child psychiatrist who work in close collaboration with us in addition to treating patients to provide the samples. And I don't have here, but the genetic studies are done in collaboration with basically Hakon Hakonarsson and Michael Gonzalez at CHOP. Obviously, we're very grateful to the funding from the NIMH for the mouse studies, to the International OCD Foundation for the initial seed pilot funding to do the cytokine study, and then obviously to PANDAS network for supporting continuing our efforts to do the cytokine study and the human genetic studies. And obviously Newport Equities for giving us the funding, the seed funding initially to really start all this work. Without that, this would have never been possible. And with that, I'll conclude, and I'm happy to take questions.
Thank you. That was terrific. And we especially appreciate your sharing so much of the research of yourself and your entire team. That was really wonderful. Thank you.
Thank you.
We do have quite a few questions. We probably will not get through all of them. We will keep a copy of the chat afterwards, and we'll try to address as many of those via email as possible. If you do want to add your email address in the chat so that we can respond to you if we don't have time here, that would be terrific. I'll start with a I think important question in terms of where we are in the field. Given what you've presented today and the other research you participated in, do you feel we're past the point of controversy about this disease mechanism existing?
I hope so. I hope so. I mean, I think that, particularly I feel confident that I think there should be no controversy that indeed PANDAS and PANS is neuroinflammatory in origin. Whether indeed is an autoimmune, it's still a little questionable because obviously there is quite a bit of diversity in terms of the presence or absence of antibodies. But basically, with both the antibodies and the identification recently of T cells targeting the dopaminergic receptors from Russell-Tail, I think there is very clear evidence that indeed there is a very strong neuroinflammatory and potentially autoimmune component on the disease. Obviously, from our data, and I think also from Jennifer Frankovich, the genetic studies, she has found that there is a great association with the HLA type 1, and we found association with HLA type 2, and also a lot of the immune genes. I think that this should be a really clear evidence that there is a very strong immune component driving this disease.
That's great. And do you feel like the medical community, that the doctors now understand that there's not a controversy, or is there still work to be done there?
I think there is a lot of work to be done here. Unfortunately, I had a recent encounter with this, basically, I think the controversy that exists with disease from a seminar that was given at our very own institution from a colleague from the West Coast, where basically it was very obvious that a lot of the physicians are still unaware of the current studies that are going on. And obviously, some of these are unpublished, but I do think that, we as researchers and we as scientists, both physicians and basic scientists, have an obligation to really kind of push ourselves to understand disease mechanisms, because otherwise it's going to be impossible to treat the children. And I think for someone to not even acknowledge the fact that indeed there may potentially be neuroimmune mechanisms underlying the disease, I think can be detrimental and potentially harmful to the children.
Okay, thank you very much. All right, we have a number of questions. I'm going to give you just a few of them here. We have one person who asks, aside from acute infectious states, do PANDAS and PANS patients tend to continue to demonstrate elevated strep levels over the years? And if so, what is the ongoing risk of those inflammatory activities and ramifications for healthcare? Should they be addressed in some manner?
Oh, sorry, it went very fast, and so maybe we can break the question in a couple of mini questions.
All right, so I think the main question is, do they continue to demonstrate elevated strep levels over the years? Or I suppose more accurately, do they continue to demonstrate inflammatory activities?
So, I mean, I think that's an important question, whether indeed there is still, I mean, in our animal studies, it's very clear that you do need multiple infections with group A strep to develop this disease. But I think that, once you have more than 4 infections, those T cells are very high numbers in the brain. And so potentially in the future, you don't necessarily need to have group A strep infections to basically trigger an activation of the disease state. And obviously, this remains a big question, but there might be a point of transition where you move potentially from an infectious to an inflammatory disease, to more a chronic inflammatory disease. And I think that that's something that potentially could be interesting to study in the future. Obviously, in the cases that we analyzed, we didn't really address that. We really were focused on the acute phase of the disease. A lot of these children at the time of the presentation, at least from the Columbia, where we actually do a swab and we culture it in the lab, don't appear to have an active group A strep infection. So, if you're going by that criteria, you may say, well, it's not, we weren't able to isolate the bacteria, but it doesn't mean that they don't have an active inflammation going on. And so I think that, considering how you treat this it's beyond obviously expertise, but I think one has to consider a potentially chronic state of neuroinflammation that is present in these children.
Okay, great, thank you. I have a question here about how long before we may see biomarkers being published, and why it's such a challenge to find biomarkers for PANDAS and PANS, and whether other diseases always have biomarkers.
So, Alzheimer's has been around for a long time, and there's still a lot of controversy about the biomarkers for disease. I mean, even to this day, whether, A beta and tau are really markers of disease or not, it's still controversy. So I think the controversy about biomarkers exists in all the disease. We are basically, obviously pushing hard to get the cytokine paper out right now. I think our recent realization of the similarities between the mouse model and the human data put us in a hold of getting the human data out. But one of the things we have promised to the community is that obviously the moment we're submitting the paper, we're also going to put the paper in the bioarchive, which is a system where now you can make the data available to anyone, pre-publication, so that even though it's not peer reviewed, it allows for the community to really share the paper and the publication. And that's definitely something that we are going to do for this, because we do feel it's really needed for the medical community. At the same time, we are just finalizing our genetic studies, so we're hoping to submit that for a peer review relatively soon, because we do also feel that is an important, not necessarily for the parents to go to the physician and say, hey, can you test me for this gene? Because obviously we're finding there are a lot of genes, but just to give a sense that from the genetic perspective, there is a very clear relationship between the immune that these genes affect the immune responses.
Okay, great, thank you. It's a very complicated question. I appreciate your referencing back to Alzheimer's. All right, so we have just a few minutes left. I'll ask you one last question. I always like to ask this when I'm presenting. Given where we are in the field today, if I were to ask you back to present again in 3 years, what are you hoping to be able to talk about?
I am hoping to talk a lot about One of the things that we're very intrigued about in terms of understanding, obviously, is expanding the genetic study. And I think that one of the things that I would love to see is if we were to do this in a large, cohort, will we find the same genes or will we find something different? And that's one of the things that I am very intrigued about. Obviously, in that context, we're also intrigued in terms of looking at potentially these genes that we identified. What is their function, in the CNS? And I think that obviously our animal studies are going to be able to address that. But I also want to think a little bit more broadly about bridging the gap between autoimmune encephalitis and post-infectious basal ganglia encephalitis, and potentially setting collaborations with experts. Some of those are here in the list of attendees who have expertise on autoimmune encephalitis, and potentially how we can think about bridging the gaps or really thinking broadly about these disease entities and how we can address mechanisms that are similarity. I mean, there was some beautiful paper published by Gary Westbrook showing that T cells are also required in an animal model of autoimmune encephalitis. So to me, that paper really speaks very clearly that indeed these cells play an important role in these disease entities. And I think obviously bridging that gap would be really important for the entire field and the community. Because obviously, I think, there are very clear treatment guidelines for autoimmune encephalitis that exist right now. And also it's a disease that I think it's covered by, the treatment is covered by insurance. That's not the case for PANDAS and PANS. And I think that by really bridging this gap, we might be able to, improve the treatment for children, obviously.
I really appreciate your passion for helping the families that are impacted by this. Your work has been really influential, and we're all really deeply grateful. I hope many of you here on this call will end up collaborating, and then we'll hear about your research. Yes, reach out.
Yeah.
That wraps us up here. I'm just going to share again our contact information in case you'd like to send questions to us or have questions about treatment. We will try to do additional webinars on the treatment side of things for those of you that have questions in relation to that area. Again, we deeply appreciate your being here with us today, and hope that we'll see you again.
Susan, can I just add one more thing? So for those of you who I wasn't able to answer the questions, I'm sure that Vicky can send me the questions and I can answer. But if you want to ask those questions directly, you can email me, and I'll be happy to respond to the best of my knowledge. The email is da191@comc.columbia.edu, but you can find me online.
Thank you for being so accessible. It's really appreciated.
Thank you.
All right, we will be winding up our seminar now. Thank you so much for all of you who participated. And as a reminder, we will send out information on accreditation right after this seminar. Thank you so much. Have a great day.